In this video, we will discuss about this microcontact anti-CD3 with equal surface density on different stiffness of hydrogel. So they have optimized their protocol to have equal surface density on different stiffness of hydrogels of this anti-CD3. And thereafter, what they did is they measure their results with a control in which they printed the anti-CD3 using a flat stem, whereas in the microcontact anti-CD3 printing they used a circular pattern, and then they check the intensity, fluorescence intensity at different positions, and one of their examples is shown in this panel ABC, where we can see that whenever they identified a dot, there is an increase in the fluorescence, and then there is a high contrast between the background and the anti-CD3 conjugated to PE anti-mouse antibodies. So we can see that there is a high contrast between the presence of dot and the background, and the optimized diameter of this anti-CD3 that they have identified using the literatures and also during their hydrogel preparations is 8 micrometer. So here the optimized results are shown, where we can see that the fluorescence intensity is almost comparable for different stiffness of the hydrogels, and these things are also quantified in this panel D, where we can see that the mean fluorescence intensity within the spots is comparable and it's less than control. And when they did the experiment for the where they print anti-CD3 using flat stem, they are able to see the fluorescence intensity, and this is in the Z directions, and these things are quantified in the panel F, where we can see that mean fluorescence intensity is almost comparable for the different stiffness of the hydrogels, but for the control there is no fluorescence. It indicates that this mean fluorescence intensity are the result of the presence of anti-CD3 which is conjugated to PE anti-mouse and PE anti-mouse, and there are many supplemental figures where they have shown how they have optimized their protocols to get this comparable fluorescence intensity for the different stiffness of the hydrogels, and this is essential for their studies further, because they don't want to include any more additional variables to taken into account while understanding the interactions of the T cells with the T cell receptors, that is anti-CD3, that can trigger the signaling pathways within the cells. And their final aim is to obtain the mechanical interactions between the antigen presenting cells and the T cells, and what are the signaling pathways that get triggered when the T cells get activated by T cell receptors, that is anti-CD3. And they will measure it on different stiffness, because when the cancers get developed within the body, it will also change its surrounding environment, and it will form a tumor microenvironment which has a complete different characteristics, and one of the characteristics that varies is the stiffness around the tumors, that will also play a role towards the proliferations and uncontrolled growth rate in the pancreatic cancer cells, at the same time prevent them from the immune detections or helps them in the immune evasion, which is one of the important aspect of the cancers if they want to survive. I hope you understood how they did their macro contact anti-CD3 how they developed their microcontact anti-CD3 with equal surface density on different stiffness of hydrogens. If you need to understand more about their experimental approach in detail, you can go through the reference where you can find supplemental figures to understand their understand this concept in a better manner. So that's all for this video. Thank you for your time.